Hi, everyone. Welcome to today's webinar, Move Forward with Automation. I'm Casey Ganchert from Eppendorf, North America's Manual Liquid Handling team, and I'll be your moderator for today's event. We are so excited you could join us this morning. Today's educational webinar is presented by LabRoots and brought to you by Eppendorf. To learn more, you can visit eppendorf.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want throughout the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to present today's webinar speakers, Paulina Kocjan and Kirk Mantione from Eppendorf, North America's Automated Liquid Handling team. With that, I'm now going to turn the stage to Paulina.
Thank you so much, Casey, for the introduction. So hi, everyone. Thank you again for being here today. In this webinar, we're going to be talking about the benefits of automation and how to decide on a platform that is best fit for your application. So why automation? What are the true benefits of automating your workflow, or even a part of your workflow for that matter? As you may know, most workflows in the lab require you to handle different types of liquids. With manual pipetting, the largest source of error is actually due to the person holding the pipette in their hand. The operator accounts for 70% of the source of error with manual pipetting. All the other factors listed, such as the actual pipette being used and the type of liquid being transferred, additionally contribute to variability with manual pipetting. Operator error can be due to the pipetting technique being used, which typically varies from user to user, and having distractions in the lab, which may cause mistakes while pipetting. When using a pipette in the lab, you may not be aware of the numerous pipetting best practices. Are you conscious of how far down you are pipetting into the well of your sample plate? In Eppendorf's Liquid Handling Guide, the proper pipetting technique is described, including optimal angle and immersion depth when pipetting. Scientists or lab technicians may additionally have other priorities on their mind, such as meetings to get to or working on analyzing their data. When pipetting in the lab, they may also have distractions that could impact the reproducibility and reliability of their results. For example, a lab technician may be talking to a colleague in the lab, which could cause them to forget what well they were actually pipetting into. So based on your experience pipetting in the lab, do you think you could get these results manually? Our automation platform, the Eppendorf EP Motion, tested the transfer of 1 microliter using 40 different pipetting tools. You can see the amount of control of an automated liquid handler from one tool to the next. The error bars in the graph represent 10 transfers from each individual pipetting tool. So we talked a bit about user error, but now let's dive into pipetting of difficult liquids. Based on your experience, have you ever seen droplets when trying to pipette a troublesome liquid? If so, what have you done manually to ensure an accurate transfer? Droplets form when liquids with a high vapor pressure, such as acetone and ethanol, force liquid out of the tip. These types of liquids can be difficult to transfer manually. One technique to reduce or even stop the dripping is to achieve a higher percentage of humidity in the air cushion inside the pipette. This is done by pre-wetting the pipette tip a minimum of 3 times, which saturates the air cushion for an accurate transfer. However, at very high concentrations, pre-wetting the pipette will not be enough. In this case, it's best to use a positive displacement pipette. Positive displacement pipettes use tips with an integrated piston, eliminating the risk of dripping. Viscous liquids are another example of challenging liquids. Due to their thick consistency, viscous liquids do not flow well and tend to get stuck in the tip of the pipette. Your best option to combat this problem is to pipette slowly and wait for the liquid to dispense. You can optimize your manual liquid handling by adjusting pipetting speed, waiting for the liquid to move down inside of the pipette tip, or by touching the pipette tip to the edge of the well. Many liquid transfers like this take trial and error to ensure accurate and precise results. Similar techniques can be used on an automated platform to achieve accurate transfers, especially for successful miniaturization in applications such as PCR or NGS. In the table shown, there is a CV of 0.6% and relative inaccuracy of 1% when automating the transfer of 1 microliter 24 times on the Eppendorf EP motion. The CV for the transfer of 0.2 microliters is at 1.8%, which is very precise liquid handling. This is the type of accuracy and precision you can expect with high-quality automation. Now thinking back to the user, is manually pipetting also limiting their productivity in the lab? When pipetting, the user may not have time to focus on anything other than transferring liquids, making tasks like data analysis and additional research a second priority. Pipetting can be time-consuming, and let's be honest, we also all have different responsibilities outside of work. Automation can increase your productivity by helping you with routine pipetting tasks that can often be quite tedious. Depending on your throughput needs, you may want to run 8 to 96 samples at a time. The user can run a few samples or even an entire month's worth of samples while working at their desk. Keep in mind that it's not always about speed, but overall productivity when we're talking about the benefits of automation. With increased throughput, sample traceability throughout your workflow can also become difficult. Automated systems can assist you by providing reliable data management such as liquid transfer data, error logging, and sample information. A good liquid handling platform should either seamlessly integrate with a LIMS system or have the capability to export liquid transfer data. The barcode on the consumable allows for the complete traceability of the tube or the plate containing your sample. So now that we have discussed some of the benefits of automation, let's dive into understanding your choices and how to find the right level of automation for you. So how sophisticated of a robot does your lab actually need? So what are the major concerns when it comes to mind thinking about automation? What makes it so difficult to commit to an automation platform? Typically, one of the biggest concerns is the large learning curve. Your thought process may be, will I need to learn a new language? Will I need programming experience? I don't want to waste time learning something I will never use. You probably start thinking about how much time you'll want to put into learning the software of a new automation platform. If it takes too long to use, you may think it's better to run a process by hand. Automation can be scary. Software bugs may come up that can make it difficult to recover from, leaving you at a stopping point. I remember my first crash when I started in automation, and it made me so nervous. The multi-channel head crashed into a box of tips that the system didn't know about. The head was stuck, and I needed to somehow recover. And of course, at this moment, I was in panic, but I decided I stopped the system, I reinitialized it, and the head was no longer stuck. So I was able to jog the head back into a normal position. Once I learned how to recover and avoid such crashes, I became very comfortable with automation. I can understand the need to feel confident to automate your protocol or method. You want to be in control of the protocol you are running and in turn, ensure you're getting the results when you need them. It can also seem challenging to transition from manual processes to automation. Not only do you have to think about liquid transfers, but also about the deck configuration, tip usage, and the details of all of your pipetting steps, including volumes, air gaps, and flow rates. These concerns are realistic, but depending on the automation platform you choose, they can be somewhat alleviated. An additional concern could be around the level of software flexibility your lab needs. Once a protocol is finalized on your liquid handler, are you now locked into that protocol? Are you able to make edits as needed? Are you able to quickly write a new method? Depending on the liquid handler you choose, it may be difficult to optimize your process. You'll probably need to implement new labware at some point that has to be verified on the system before using it in your automated protocol. You'll also need to optimize your liquid transfers and tip in reagent usage for your application. For example, in NGS bead cleanups, you may want to reuse tips for your ethanol washes and mix beads with your samples a certain number of times before going onto the magnet. These are the types of optimizations that you will probably want to do on your own. We now discuss some valid concerns, which are all important to keep in mind when selecting a liquid handler that is best fit for you. So how do you then go about choosing the right platform? There are a few questions that can be good to keep in mind when considering automation. These questions can be used as a framework to help you make an informed decision. The first thing to consider is your sample throughput. So what is throughput? And will your throughput grow over time? Throughput can be the number of plates produced per week or the number of samples run in a day. Understanding your level of throughput will help you decide what liquid handler is best fit for you. The second thing to consider is the level of automation expertise in your lab. If your team doesn't include automation engineers or experts, your lab will probably need a user-friendly software. Turnover is inevitable, making it very likely that a new person will need to be trained on the system. Taking it a step further, how many people will be using the system? The more people that will be using the liquid handler in the lab means more methods that need to be written. So this means that it's extremely important to think about the usability of the software of the liquid handler you are considering. Another consideration when choosing an automated liquid handler is platform size. Lab bench space is very important. So how much are you willing to give up? Does the system need any additional connections, such as water, electric, or air supply? Will this mean you'll need to get in touch with facilities in order to get this liquid handler set up in your lab? The feasibility of accessing these additional connections may alter the liquid handler that is best fit for you. The final consideration is deciding on how much of your process should and can be automated. Larger liquid handlers can typically automate all steps with additional device integration. But for example, is it a deal breaker for you to carry your sample plates to a thermocycler? Or does your lab really need that thermocycler on deck? That is definitely something to think about. The level of integration and automation that your lab needs will influence your final automation decision. Now that we have reviewed what questions to ask when choosing an automation platform, there are a few key topics that I think you should remember. First, the system should meet the needs of flexibility in terms of customization, such as method changes and the use of different applications. The software should allow you to easily adjust as needed. Additionally, if your lab requires full walk-away capabilities, a liquid handler should be able to integrate with devices such as thermocyclers, plate washers, and readers. Depending on the automation platform you choose, there should be an appropriate level of application support or expertise. Once you have all of your questions answered, you should feel comfortable on deciding what your automation platform should be for your lab. I will now be passing the stage to Kirk Mantione, who will talk to you about what your solution can look like, as well as give you some examples of some commonly automated methods.
Thank you, Paulina. Hi, everybody. Thanks for taking the time out of your day today to hear a little bit about automation and what your automation solution could look like. So Paulina just spent a good amount of time talking about the questions that you need to ask to decide on what kind of automation solution you might use. So part of my job at Eppendorf, about half of my time is spent helping people choose the right automation platform. So depending on all those questions we just posed, you might have more questions, and that's where the automation specialist would help you answer the rest of those questions. And then we'll be able to show you what an easy solution for your workflow might look like. So Casey and Paulina were discussing what automation might look like for you and what kind of issues people have that might make them resist trying to automate. So one of the things we come across most of the time is the lack of programming experience. So do they really need to know how to write in a particular code for a computer, or do they need some more information? Do they need how to measure things and program things? So a good automation system, you don't need any programming experience. You don't need any more experience than being able to use a computer, whether it's Windows or Mac-based computer. So as long as you can do that, a good automation system should be a good fit for you. So what you really need to understand is what you're doing in the lab, the science part of it. So we'll leave the science to the people in the lab and leave the automation to the people that make the automated platform. So if you understand what you're doing by hand and creating and setting up reactions manually, the robot should be able to do what you're doing by hand. So another resistance to automating that people have is, I don't want to convert all my manual liquid handling steps onto a robot. It seems like it would be very difficult to do. But what I'm here to tell you is that if you have a good platform that it's easy to use the software, you can simply tell the robot what you're doing step by step and transfer liquid from point A to point B, because that's really all liquid handler is doing, the same thing you would be doing by hand. You're transferring liquid from point A to point B, and we can all do that. I've talked to some of our salespeople at Eppendorf, and they help people to show them like how to automate it, how you would automate a particular method. And sometimes the customers would give us paper protocols and say, all right, here's what I do on paper, this is what I do, and this is our standard operating procedure. And sometimes that's great because we can just simply write step for step what they do. But this one person that we had, a salesperson that we had, would say, don't give me a piece of paper, just tell me what you're doing, and I'll make the robot do what you're doing by hand. So it should be that easy for you, and it's much easier when you just have to tell somebody what to do and program it, and that's what automation specialists do at Eppendorf. So the protocol changes should also be easy. So if you want to, for example, I'm pipetting 8 samples today, and tomorrow I want to do 96 samples. Those changes should be easy, and anyone in the lab should be able to make those changes on the fly. And that's what a good automation system should also have. So what we're doing is we're basically taking the pipette out of your hands and putting it onto a robot, and you're free to do other tasks while the robot is working with you. You're free to watch the robot if you want. I know a lot of people that when they first get systems, they sit there and they watch the robot because it's fascinating to watch a robot do its thing. But we encourage you to do other things in the lab besides watch the robot. This next slide is about the software usability. And good liquid handlers, as I was saying, you don't need to know about coding. You just want to tell it to pipette from A to B. So these are some examples of the software on the EP Motion. And there's, on the top line, there are commands for liquid transfer. And liquid transfers are exactly that. I want to transfer these 10 samples into those 20 wells, or I want to add a bulk reagent across an entire plate. So, or I want to dilute a sample, or I want to pool a sample into one tube. Those are all things that if you've worked in a laboratory, you've used those terms before, and the software uses those same terms. So it's pretty easy to understand. I've been with Eppendorf for 3 years, and I didn't have any automation experience. I came from a molecular biology lab. And for me, it took me about a week or two, and I was able to quickly program methods, and now I'm programming methods for other people. So the learning curve is not that steep, and once you start doing it on your own, you'll find that you want to automate everything that you've been doing in your lab. The other commands on there that are fairly straightforward to understand, are things that you do anyway in the lab. You want to mix by pipette, or you want to shake something, or heat or cool something, or wait at a particular step. These are all the other auxiliary commands that are offered on most good liquid handlers, and they're just as easily as dragging and dropping. So to program these type of liquid handlers, it's just a matter of dragging and dropping the sequence of the commands that you want. The other objection that a lot of people have to automation is the flexibility of an automated liquid handler. And the Eppendorf system is called an open platform. And what that means is you can use many different other manufacturers' plates and tubes and use them on the same platform. So you can, for example, you have a bunch of Eppendorf plates and you run out, but you have a shelf full of plates from another manufacturer. You can quickly grab them and use them on the platform. And you don't need to know anything about coding that particular plate. All you need to know is what's the part number of that particular plate. The software has those part numbers preloaded in it, and it's a matter of dragging and dropping the new plate onto this virtual work table. Another good thing to look for in an automated liquid handler is how easy the software helps you out. If you're not familiar with programming and you're new to the system, you want a software that can check for logical errors or alert you of possible crashes. As Paulina was talking about, she had that crash that the pipette tool didn't know that there were tips on the deck and it crashed. So some liquid handlers have the ability to scan the deck before you even start the system. It will scan and it'll look for tips, it'll count the tips, it'll look for the plates that you're using, it'll look and check the volume of reagents, make sure you have the right amount of reagents on there. So that's another good thing to look for when you're shopping around for liquid handlers. You want to be able to minimize errors right away, and you want the software to help you program correctly. The other thing that we have available would be verifying the liquid volumes. And as I said, it can tell you you don't have enough liquid on the deck, so you would know to add more liquid for a particular reagent. Now I want to transition into a little bit of, besides the actual hardware that a liquid handling company might offer, the support that they're going to offer you. We're spending a lot of time saying, yes, these methods and this writing of methods are easy and simple and anyone can do it, but we all know how limited we are in time. So we do offer, and good liquid handling companies offer, application support. And as you're shopping around and as you're trying to automate a method, you should look for companies and kits. Ask the kit manufacturer, hey, has anyone automated this kit before on any systems? They more than likely will have a list of the systems that they've been automated on already and successfully generated data. So that's another thing you want to look for. Definitely ask the kit manufacturer or ask the liquid handling manufacturer, have they automated it before? That's a great place to start. The other benefit of using application support, besides shopping around and helping you pick the right platform, is once you do decide on a platform, that they're going to be available, whether it's virtually or coming on site, to help you to maximize the efficiency of the robot, to minimize the consumable usage, and to get you started right away, so that they'll come in, program the method, test the method with you, train your staff, and present you with a user guide after their visit is over. So a user guide is great because you can incorporate that into your standard operating procedure, and it makes it easy for other people in the lab to be able to use the system. So you might have more than one method, you might have 10 methods on a system, and that's important to be able to just pull from a file and say, this is how you set up this particular method. So what we do at Eppendorf is we provide you with a user guide after we come in and program a robot. The user guide has everything on it that you would need to just grab off the shelf, put it on the deck, and it kind of makes it plug and play. You know how much volume of a reagent to put on there, you know which consumables to use. And I like to use the analogy is if you had a lab tech parachute into your lab and suddenly they've worked in other labs before, so they're familiar with laboratory practices, they could grab the user guide, grab the robot, and program it and get it up and running without having any learning curve or any time spent figuring out what to do. So that's one of the great benefits of having application support and a good user guide. So now I'm going to transition and talk about some of the methods that can be automated on liquid handlers. And these are just some frequently used methods, and most of the time, because I come from a molecular bio background, I'm going to talk about some molecular biological methods that we've frequently automated. And as Paulina's part of the talk, she talked about all the benefits of automation, whether it's the efficiency of it, whether it's the accuracy and precision. All these things tie into these methods and why these methods are great candidates for automation. So the first one we're going to bring up is the polymerase chain reaction. Whether it's going to be your conventional PCR or your gel-based PCR or your real-time PCR, it's all the same reaction setup, right? We're adding our master mixes to tubes, we're adding our templates to tubes, and we're bringing them to a thermocycler. So setting up conventional or real-time PCR, we have the same challenges. We've all gotten blank gels or no amplification curves, and we go back to the drawing board and figure out what went wrong. Now I have to repeat an experiment. So automation is taking out one of the variables, that variable being human error. Did I make a mistake? Did I forget to add that reagent? So if we can take out one of those variables, that's one of the benefits of automation. The variables are reduced and it makes it easier to troubleshoot. The next slide is a list of the issues that we frequently run into while we're setting up PCR. We all know it's a highly sensitive reaction, so volume differences make a huge difference in the outcome. It's a tedious process where we're typically running, if we're doing real-time PCR, we're running reactions in triplicate or quadruplicate. So there's many reactions that have to get set up. And the bottom line is the bottom line here. It's expensive. The reagents are expensive. And if you have to repeat an experiment, now you're burning through reagents and consumables. We don't want to have to repeat things. We want to get it right the first time. You can see here, highly sensitive. This is an example of an amplification curve. And each one of these colored lines represents a quadruplicate reaction. And this was set up on an automated platform. And you can see where these lines cross the threshold are all very precise. So we want to have that precision, and this precision is over 6 orders of magnitude. So this shows you that you automate it, you can get highly sensitive reactions, and you can get them reproducibly. The next thing, we've all tried to pipette. I know I used to just do 96 well plates, but we're all trying to scale down reactions. It's a 384 well plate. Paulina talked about distractions in the laboratory. I try to pipette into well B1, and I get tapped on the shoulder, and it's my PI asking me if I have that data yet. And I tell him, oh yes, I'll be right with you. And then I look back at my plate and I say, oh, did I add to B1 or did I add to B2? So if you have a robot doing this, you can talk to your PI or you can talk to your colleagues, and the robot is doing the pipetting. So take that human error out of the equation. So these are some of the solutions that we offer. We talked about why automation is so good, and I think that PCR is probably one of the best illustrations of how you can benefit from automation. So why wouldn't you want to automate? PCR is one of the best methods to illustrate it. You pipette in the same well every time. You're going to have a reduced risk of cross-contamination because of some of the controls that are provided by some of the liquid handlers and the tools. You're not going to have drips across the deck, and it makes it easy to miniaturize reactions. You're not going to have to worry about that tedium of miniaturized reactions. And I'm just going to show one more slide with some data that was generated on an automated liquid handler. This is some data that was generated on an EP Motion. On the left of the slide is the deck layout, and this is just the simplest reaction. We've done reactions with either 50 microliters or 10 microliters, and the gel pictures on the left show you that you get the same basic yield and same brightness of the gel bands when you're scaling down reactions to show you that the results are comparable and reproducible, whether you're doing it by hand or on a liquid handler. The next method I want to talk about is nucleic acid purification. Again, any molecular biology lab knows that isolating DNA or RNA is always the first step in most of the protocols. And you always want to get high quality nucleic acids and make sure that the sample handling is not introducing an artifact. It's probably one of the most time-consuming steps in the process of getting your nucleic acids. It has the biggest risk of cross-contamination if you're working in 96-well plates, or even if you're working in individual tubes. You always run the risk of, did I add to that tube, or did I change tips? Was I supposed to change tips there? So the robot is going to be able to know what to do, and as long as you program it correctly, you're going to reduce the risk of cross-contamination. And you're also going to be highly consistent. You're not going to make errors, because as long as you program the robot correctly, the errors will be minimized. So difficult pipetting steps is the next slide, and this is important whether you're using a nucleic acid preparation that requires a filter column, or you're doing magnetic bead-based nucleic acid prep. The pipetting height needs to be exact, and trying to do this by hand, some people have shaky hands, they might poke through the membrane of a filter column, or you might slurp up some beads that aren't supposed to be slurped up. So with an automated liquid handler, you can set that height exactly, and it's going to do the same height every time. So once you optimize it, the method's locked in, and it's not going to change. It's going to be very, very consistent. The next slide just shows an example of, on the left is how easy it is to control an automated liquid handler that has a vacuum manifold. You just set your time of the vacuum and the pressure on the vacuum, and most of the time, kit manufacturers will have that number already told to you, and you just dial it in and test it with some water and make sure that the timing is right, and it pulls the vacuum and pulls your sample through in the right amount of time. And on the left, we just have an illustration of a 96-place magnet with a 96-well plate on it, just pulling down some beads. You get that nice ring, and you come in with your pipette tip, and you can remove liquid in the same spot every time. And nucleic acid preparation on liquid handlers, the good thing about it is on some medium size or small size liquid handlers, you can run up to 96 samples at a time. You're definitely going to have the reduced risk of contamination, and you're going to get highly reproducible results. So one more slide with some data. This is some data from a blood isolation kit that shows you that you can get consistent and pure nucleic acids, automating it on a liquid handler. And on the right, it just shows you can put all your reagents on the deck, and you can be able to get everything done at the same time. Another thing that obviously most of the world has been doing right now has been RNA isolation from viral material. And these methods have been routinely automated on many different liquid handlers from many different companies around the world for SARS-CoV-2 research for obvious reasons. So just to let you know that this is being done, and it's being done very efficiently right now. And the final method that I'm going to talk about is next generation sequencing and preparation of libraries for NGS. So it's a very expensive process, very tedious, very time consuming, and there are many steps involved. It's not terribly complicated. I like to say it's a simple method, but it's an elegant method. So all these steps have been worked out for us, and kit manufacturers provide you with everything you need to do all these steps, from sample collection to library prep to quality control to sequencing. Each have many different sub-methods, and these sub-methods might be PCR, which we talked about before, and we know we can automate that. Setting up normalization liquid handling, that can be done on a liquid handler. Bead cleanups, we showed you we can do it for nucleic acid prep, but you can also do it for size selection of libraries. So all of these methods, or one of these methods, or all of them, can be automated on good liquid handlers. So you don't have to automate the whole thing, but you can automate the ones that are most challenging to you, or the ones that you really want to automate. You're not locked in. So this is some example data from what was formerly known as the Nextera Flex, now known as the Illumina DNA Prep Kit. And this is one of the most common methods that we've automated. This is a data set from a laboratory that I worked in Canada. They were nice enough to share the data with me from their tape station, just to show you that you're getting the size and you're getting the right size selection and the right purity without any primer dimers or anything like that, or some leftover material. It looks pure and clean. The next slide is just, most of the time before we automate anything, we want to do it by hand. If you can't do a method by hand, don't attempt to automate it. So this is what we'd like to do when I go into somebody's laboratory and I automate a method for them. I'm always going to compare it to what they got by hand. And this is from one of the Eppendorf's application notes, where we went in and did it alongside our kit manufacturing partner and a customer's laboratory, where we generated this real data set. And just this shows you how close they are to being, how close together they are. So most people are very comfortable doing things manually and are apprehensive to automating them. This shows you that you automate this particular kit and you can get comparable results, the same yield and the same callability. And the summary for the NGS is what we talk about anytime we talk about automation: a low failure rate. You don't want to have to redo reactions. You want to automate as many pipetting steps as you can to take out that human error, and you want it to be scalable and consistent. Whether you're going to set up 8 libraries or 88 libraries, it should give you the same results every time. And to summarize everything we talked about today, tips for successful automation and selecting the right automation platform. You always want to ask as many questions as you can. You want to know what application you're doing, and know that if you're doing more than one application, select a robot that can easily switch between the 2 applications. There are many robots out there that might just have one type of application built in, and you don't have the flexibility. You want to look for something that's flexible if you need that flexibility. You want to make sure that all your steps are available to do, whether it's heating, cooling, and shaking. You might want to do things hands-on, or you might want to do them on the robot. And automation is not always about speed. You might have a library prep that takes you 4 hours by hand. It might take you 4 hours on the robot, but that's 4 hours that you're not spending hovering over your samples. You're doing something else in the lab besides pipetting. And always look for kits that can easily be optimized. Sometimes kits don't have an exact match for automation, and you might have to adjust it. So bear in mind that you might have to be flexible with your concerns and flexible with what you normally do, because it might not be a perfect fit all the time. So look for that. And as long as you pick the right fit, automation should be easy, and it shouldn't be any stress about it. And now we're going to open up the floor to some questions. If anyone has any questions, I know you've probably been putting them in the chat, but Casey will then moderate the questions and hand them out to Paulina and myself. Thank you, everybody, for your time.
Thanks, Kirk and Paulina, for both of your informative presentations. As Kirk mentioned, we're now going to start the live Q&A portion of the webinar. So as he mentioned before, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the left of your screen, and we'll answer as many questions as we can with the time that we have today. So let's get started. Our first question is, what volume ranges can the EP Motion pipette?
I can answer that one, Casey. So the EP Motion pipetting tools, there's 4 different tools that span the range from 0.2 microliters all the way up to 1 ml.
Perfect. So we have another question. How long does it typically take to get something automated?
Yeah, sure. So I think this really depends on the automation platform you choose. There's time that's needed for installation, service, and you need applications time for training and protocol development. Really, this is where the usability of the software comes in, and it's an important factor in automating your methods and how quickly and how easily that can be done.
Great, thank you, Paulina. So we have another question here. How would you normalize your samples on the EP motion?
I would answer that one. That one is a frequently asked question that I get, and typically on the EP motion, you would need to create a CSV file so you can make tables in Excel spreadsheet and import your volumes and set a series of sample transfer commands to do that. So if you wanted to, for example, normalize your concentrations, you have to measure the concentrations first. Whether you set that up on the EP motion or not is up to you. But once you know the concentrations, you then create two different tables, one with the volume of the diluent and one with the volumes of the stock solution that you want to dilute, and then import those two CSV files, and the system will generate all the sample commands for you to do that.
Awesome, thank you, Kirk. Another question, what applications are best suited for automation?
Yeah, sure, I could take this one. Thank you, Casey. Automation is really suitable for a wide variety of applications, and typically these have repetitive or time-consuming steps or require accuracy and precision, and also better sample management. So NGS is a good example. You'll see a kind of a lot of automation in that kind of world because it tends to be a process that is labor-intensive and includes multiple repetitive steps with the bead cleanup steps. So, yeah.
Thank you, Paulina. And another question here, what kind of accessories are on an automated platform?
Okay, that's a fairly open question. So accessories, when I think of accessories, I think of not only the things that can be moved on and off of the deck, like for example, a 96-place magnet would be an accessory, or you can have things that are already part of the deck, like a mixer or a heater or a shaker. Some systems offer a little bit more things than that, like some systems might offer a thermocycler on the deck. Some systems might have a tip tower on the deck. So depending on what you want, there are some accessories that can be removed from the deck, and then there are some accessories that are built into a deck. So that depends on the system that you're looking at, but those are the type of things that we consider accessories.
Awesome. And I think you touched on this earlier, Kirk, but there was a question that came through about the minimum volume that the EP motion can pipette. You want to touch on that?
All right. So the minimum volume that the EP motion can handle is 0.2 microliters. That's with our tool that has a range from 0.2 to 10 microliters. Does that make sense?
One more question here. Is there a chance of inaccuracy with automation using a software, and is there a chance of malfunction with the programming?
Right. So, kind of like we talked about earlier, I think with whatever automation platform you choose, there can always be issues, right? I think in terms of the actual tool, it's important that you get it calibrated and serviced so that you know you're basically having accurate transfers. Because usually the tools, whatever pipetting heads you might be using, are calibrated before they come to you. So they should be quite accurate. You could obviously always test in-house if at all you're like questioning the accuracy of the tool. And in terms of also kind of, if you have the system for a long time, you may have O-rings that fall off or leaks or something that's not functioning, and then that would be a service. But typically you should see pretty accurate transfers. And in terms of the software, there you'll program like usual and put in the number that you actually want to transfer. So that should be easy to do. And then you shouldn't see any issues on the software side.
Awesome. Thank you, Paulina. So another question, in the steps that require incubating at high temperatures, such as 65 degrees, is there a possibility of sample loss due to evaporation? How can one avoid this?
That's a great question. I can handle that one. So on the EP motion, I have a general rule when I'm doing high temperature incubation. So I typically, anything above 40 degrees for longer than 10 or 15 minutes, I would avoid evaporation by adding mineral oil to the sample to prevent evaporation. Either that, or if it's a longer incubation on the EP motion, I would take it to a thermocycler with a heated lid.
So much, Kirk. Can you please specify the limitation of Eppendorf's EP motion compared with other solutions?
Yeah, so I think I can answer this, and maybe Kirk can, you can add on to this too. But, you know, every type of liquid handler is different based on, you know, what you're looking for. So this kind of goes back into what we talked at the very beginning. So depending on, you know, throughput is one of the really big ones, and ease of use and integration. So if you want to, you know, a system that can be extremely high throughput, and let's say have a 384 well multi-channel head, then that would be, you know, a different system than the EP motion, right? And also if you wanted to have a full walk away of integrated devices, that would also be something different. But keep in mind, usually with, you know, whatever you add on kind of to that, the platforms do become also more expensive, right? And more complicated, I'll say. But yeah.
Yeah,
that's, I would.
Yes, I agree with that. I mean, that's one of the things about the EP motion platform. We're low to medium throughput, so that's one of the limitations. If you wanted to do more than a 96 well plate for NGS, that would be the, you know, the upper limit for the EP motion platforms. The other thing would be you're limited in terms of like some of the patterns that you can create with the system. So when I was hinting at before during the talk was you have to adapt your method to the platform. So if you're used to pipetting across rows rather than down a column, that would be a limitation of the EP motion, and that's just a matter of reprogramming it and resetting your expectations.
Awesome. Thank you guys so much. We're just rolling through questions here. We have a question that came in asking if PCR is a cost-effective method.
A cost-effective method, I can take that. As far as on to set it up on to spend the money on an automated liquid handler, yes, I think it would be a cost-effective method, especially if you're doing higher amounts. If you're doing a 96 well plate a day, then it's definitely a cost-effective way to do it. And then you're not going to use any more reagents or consumables on an automated platform than you are by hand. I hope I answered that question in terms of what you're asking. I hope that wasn't a philosophical question.
Great, Kirk. Thank you so much.
All right.
So another question here as well. In software usability, what is the required temperature? Is there any required temp for software?
I'm not sure I understand this. Does that mean, I'm going to take a stab at it anyway, required temperature as far as the ambient conditions of the laboratory? There is a normal room temperature would be the range. That would be more of a question if we could, I might be able to find out more information about that in terms of what our particular, Eppendorf's particular liquid handling platform has been rated at as far as the temperature and humidity that you can operate the system in or the software in, that we could get back to you on that one.
Right, yeah. I think that's a pretty good way of answering it.
Great, guys, thank you so much. Another one. How can accuracy and precision be reached when it comes to automation?
I can handle that one too. How could it be reached? Just like you do it manually, trial and error most of the time. We have a pretty good idea of if what you're handling is something that we've handled before. We have great settings that we can provide to you and say, use these settings for that particular reagent. And then you would have to, of course, try it in reality to see if it is accurate and precise.
Right. Yeah, I think it takes optimization, but then, once you get it optimized and it's much easier to do so using the functions in the software, then you should be, once you're kind of set, you should be pretty set for whatever method you're writing. You have basically for accuracy and precision. Yeah.
Right, and that just speaks to the mindset of automating things. You spend the time up front and get everything as good as you can get it up front. I don't want to say perfect, but you get it as close to perfect as you can up front. You spend that time up front so that once you're running something routinely, you don't have to even think about it. You just press play and let it go.
Yep. Awesome. Thank you so much. And I think we have time for one last question here. Can you use a liquid handler for sterile sample dilution?
Yes, that's without, you would have to put it into a biosafety cabinet or a laminar flow hood, but there are some solutions that are offered to be able to do sterile work, but you definitely would have to have another manufacturer and obtain something to put it in to make it sterile. But we do have customers that do that.
Thank you so much. I want to say thank you again to Paulina and Kirk. And do you guys have any final comments for the audience?
Just thank you so much, for everyone for taking the time and joining us today. And we really hope you enjoyed it. And please send in any other questions, and we'll definitely get them answered to you.
Yes, I want to echo that sentiment. Thank you very much for your time today, and thank you to LabRoots for providing us with the platform to do this. All
right. Thanks again, Paulina and Kirk, for your time today. We would also like to thank LabRoots for presenting today's webinar. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any of the questions we did not have time to answer today will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. Both Eppendorf and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We'd like to thank you again for joining us today, and we hope you have a great rest of your week.